Pero ahorita te lo presento. Josh, puede
I was going to steal this. Probably.
Oh, thank you.
I would, yeah. I'll put it in my pocket.
Thanks. So a little bit.
Maybe coffee, he will not allow us to take it, but the water is okay. So we
Ah. Oh, okay. Well, it can go if we need to, but.
no, but he just was telling me.
Hmm.
Do you want me to explain a little bit of this? How do you
Sure, yeah.
use it?
Good idea.
This is on already, and with this you can.
Perfect.
Sometimes it's called on the first one, the second one, the freeze. And then there's also a laser pointer in the middle somewhere.
Very good.
I think this is.
And it's working. Okay.
Do you want me to stay here? Or can I?
I'll introduce you and we'll get started.
I'm wondering, might walk around or do you prefer that I?
Okay, well, yeah, so they're gonna, usually we use a spectrophone, but I'm pretty sure we can use this as a microphone if you want to walk around. Sí se puede usar el micrófono de aquí de cámara, ¿verdad? Okay, stop, and so, yeah, well, I'll let you with this one, then we got to turn this one on.
All right, very good. Thank you.
Buenas tardes, buenas tardes. Bienvenidos a este seminario extraordinario del Instituto de Biología. Hoy nuestro ponente es el biólogo Tyler Boggs. Tyler realizó su licenciatura en la Universidad de Cincinnati en 2017 bajo la asesoría del Dr. Joshua Gross y actualmente está concluyendo su doctorado en el mismo laboratorio. Sus principales intereses de investigación incluyen la genética evolutiva, la adaptación evolutiva, adaptación a cuevas, fisiología molecular y bioinformática. Se le ha otorgado varias becas para realizar su investigación sobre la adaptación de peces cavernícolas a bajos niveles de oxígeno y tiene 9 publicaciones describiendo su investigación. Tengo entendido que Tyler está de visita a la doctora Patricia Ornelas y le agradecemos que nos trae aquí al Instituto de Biología. El título de la plática de hoy es Survival in Bad Air, Adaptations to Low Oxygen in Mexican Cavefish. Demos la bienvenida a Tyler.
Well, hello. Thank you very much for having me. Again, my name's Tyler, and I'm from the University of Cincinnati. And today, I'm very excited to share with you some of the work that we've published and are about to publish for my dissertation. So to start, very broad, maybe. There we go. So when people think about what you need to live, the 3 most common things that people think about are things like food, water, or shelter. But there's one thing that's incredibly basic that people often overlook, and that's oxygen. We need a lot of oxygen in our air to be able to breathe. And when we think about oxygen in the environment and elsewhere, we typically think about it on 2 levels. The first being the ambient environment, how much is in the air around us, or how much oxygen is dissolved in water. And additionally, in our internal tissues, so how much oxygen do we have in our organs and circulating through our circulatory system. And I probably don't have to convince you that not having enough oxygen is very bad. And if we think about it on these 2 levels, not having enough oxygen can result in losses of biodiversity, but it can also include a lot of clinical relevance, where not having enough oxygen in our tissues can result in things like seizures, broad organ failure, and even as far as death. So how can we study, especially the evolutionary effects of not having enough oxygen, as that's unfortunately becoming more of a reality in many of the ecosystems around the world? Well, one way that we or biologists have figured out is a good way to look at this is by looking at fish. Perhaps the most famous one being the Crucian carp found in Scandinavia. Now, this fish has many very impressive adaptations, two of the most famous being that when this fish is utilizing anaerobic respiration, they're able to convert lactic acid into ethanol, which is very easily able to be removed from the body, whereas lactic acid is not. Not very many species can do this. Additionally, they have this really cool phenotype where they're able to remodel their gills depending on how much oxygen is in the water that they're swimming around in. So here on the left, you can see this gill almost looks like a sausage, some people would say. And on the right, you can see that a lot of that tissue has died and has been removed. And the effect is that they're able to increase the amount of surface area to be able to pull more oxygen into their gills from the ambient environment. Another example are certain species of cichlids from Lake Victoria in Africa. And these fish are really interesting. They do something a little bit different. So when you rear this species in low oxygen, what they're able to do is, if you notice on this picture on the right, the bars don't look exactly the same as they do on the left. This species on the right is hypoxia resistant, or they're able to live in lower amounts of oxygen. And what they do is, when they don't have enough oxygen, they switch the hemoglobin proteins that they actually use so that they can utilize more efficient proteins given the environment that they're currently in. Additionally, the same researchers from that study found out that those hemoglobins are able to more efficiently pull oxygen from the ambient environment when there's low oxygen. So not only do they switch, but they prove that it's effective, that it's beneficial for this species. And yet a third example is the evolution of the codfish, or Gadiformes. And long ago, this fish was only found in the very, very deep depths of the ocean. But over the course of 100 million years or more, this family speciated greatly. And now you find members of this family near the surface waters of the ocean. Now, what's interesting is in the deep sea, the environment is very stable. There's not very much oxygen, but it's very stable. And what they've depicted here, each one of these boxes represents a hemoglobin gene. So as this family speciated and evolved to live in more variable surface waters, they duplicated the number of hemoglobin genes over time, and they tweak them just a little bit so that they're able to more efficiently pull oxygen from a more variable environment. Now, one thing that I think is really interesting is these fish are all found in very different parts of the globe. And even in the different parts of the globe, the microhabitat and the environment that they're found in are also very different. So the Crucian carp, for example, they have to deal with very small ponds or lakes in Scandinavia that completely freeze over the winter. Now, in a very different sense, those species from Lake Victoria, they experienced, or historically their ancestors experienced, a large desiccation event. We believe it to be about 14,000 years ago. And depending upon the position that these fish were living in the lake, some of them have retained these abilities to be hypoxic resistant. And finally, the codfish we find deep in the very depths of the Pacific Ocean. However, one challenge is that the evolutionary mechanisms that led to these phenotypes is still unknown. So I believe there's room for a new model to help us understand how these changes actually came about. Now, there's another environment that we know that is also hypoxic or has lower amounts of oxygen, and that's caves. And you can do to some people, it's not boring. To me, it's not boring. but for other people, perhaps reading the Encyclopedia of Caves wouldn't be the most fun activity. But you don't even have to go that far. In fact, very recently, there's been many articles just shown in the media in the US and around the world where we see evidence that caves are low in oxygen. And we see that for a variety of reasons, even affecting the art that we find in caves. And additionally, on a little bit more of a sad note, just a few years ago, when a youth soccer team was unfortunately nearly trapped in a cave in Thailand, they almost ran out of oxygen. And so the cave was beginning to flood. But in addition to that, the speed at which they had to successfully extract these people was severely amplified because they were beginning to run out of oxygen. But while these caves are found in Europe and in Asia, we don't have to go that far to find caves. And in fact, not very far from here, in this region of northeastern Mexico that we refer to as the Sierra de Abra, we find many caves. So here what I'm showing is an image looking south on the eastern ridge of the Sierra de Abra. And if we zoomed in here, you would find lots of rivers and streams that look very fun. I would probably enjoy going there and visiting. And one of the species that we find here is this fish called Astyanax mexicanus, which will be the subject of my talk. But in addition to these rivers and streams, we also find many caves. So on this map here, all of these black dots represent individual caves, some of them connected, some of them not. And for today, I'll be focusing on 3 of those caves, Pachon, Tinaja, and Chica. And if we look at the entrances of these caves, this is a picture of the entrance to the Pachon cave, as well as Tinaja. Not quite as inviting as those nice rivers and streams that we saw. And if we were to go inside, we would find something like this. And while, again, I might find it fun to go here, other people maybe not so much. And if we were to look into these waters, we would find this fish. And although it looks very different from the fish that I showed you just a second ago, these fish are actually members of the same species. And so when we compare their environments and these 2 members of the same species, we often talk about these 2 primary differences being that caves have no light, absolutely no light. And we believe that most of the caves don't have very much food. And we often talk about how these environmental features can drive evolutionary change. And so when you just look at these 2 fish, often the phenotypes that are most commonly discussed are that the cavefish don't have eyes, they're albino, they don't have pigmentation, and they have a much slower metabolism than the surface fish do. However, again, I think it would be very interesting if we consider an additional feature of these caves, and that they don't have very much oxygen. And so one of the big questions of my dissertation is wondering if cavefish have adapted or if they have any phenotypes that may help them live in lower amounts of oxygen. So we do have some metrics that we can use. One that was actually published by Dr. Ornelas Garcia just a few years ago in Pachon, where we see a major reduction in the amount of oxygen in that cave pool compared to the terrestrial surface environment. We also have a few other metrics from around the Sierra de Abra. But if we think very broadly, Dr. Gross and I published a review on this where we investigated what the environmental features that may impact the amount of dissolved oxygen in these caves are. And when we consider how much oxygen is in water, the 2 main drivers are photosynthesis, which we don't have in caves because there's no light, as well as how well that body of water is able to mix with the atmosphere. And of course, when we're talking about caves being very, very low beneath the Earth's surface, there's an often very poor mixing with the atmosphere. So while we're missing those 2 inputs of oxygen, we also have the fish and other animals that are always respiring or using oxygen. So it can be very challenging. So again, the big question for my dissertation is, have these fish adapted to living in low oxygen? So for today, I'm going to focus on 2 main questions. or two main approaches that we've used to investigate this. The first being, are there any differences in their red blood cells? We wanted to start very broad. What would be some of the potentially easier things to see or to notice? And of course, the red blood cells being a main component of the circulatory system, we found that that might be a very logical place to begin. Additionally, we were very interested to see if there may be any differences in gene expression, or perhaps the cavefish are utilizing genes differently than surface fish do, which may confer some sort of advantage to living in low oxygen. So we'll begin by looking at some results that we've obtained by looking at their red blood cells. So the first thing that we did was we purchased this handheld instrument. Josh and I lovingly refer to it as a hemoglobinometer. And the way it works is we use these test strips and we drop a standard amount of blood on this test strip. And by using spectrophotometry, we're able to determine the amount of protein concentration of hemoglobin in the blood. So we surveyed our surface population as well as 3 cave populations. And we were very excited to find that all 3 cave populations that we surveyed had statistically higher concentrations of hemoglobin in their blood compared to the surface morph. And we were very excited by this because we didn't really have an idea if this was going to work or not. And as is often the case, when you get one answer, we had many, many more questions. And so we turned to a diagnostic tool that's often used in clinics, and that tool is called hematocrit. And so the way that this works is we collect a standard volume of blood, we wick it into very tiny capillary tubes, we spin it down, and it might be a little difficult to appreciate here, but the components of the blood will separate. So what we're able to do is we're able to establish how much of the total blood volume can be attributed to the red blood cells and not the plasma and not the white blood cells and the other components of blood. And what we found was that here the surface fish had the lowest percentage of red blood cells of their entire blood volume. And as we go up the list, the caves all seem to have a higher percentage. And again, this time we were like, okay, this makes sense now. We were working off of an assumption that the caves would be higher because they had a higher hemoglobin concentration. And like I mentioned, this is often used as a diagnostic tool in medicine. And so even if you go to your own doctor and you get blood work done, this is an often used metric that they will use to just determine how healthy you are. Now, when you have elevated hematocrit, the answer of what's underlying this is almost the same all the time. And that is that you often have more red blood cells. So they often attribute the increase in red blood cell volume to having more cells. So that was the next thing that we did. We used an instrument called a hemocytometer. It's essentially a very fancy microscope slide. And as you can see, it has these very fine laser engraved etchings. And so what we're able to do is we're able to use the distance in between those lines, as well as the distance between the slide itself and a cover slip to determine the number of red blood cells in a given volume. And so what we did was we took some blood from all of our populations. We stained it with a stain so that we could see them on the slide. And then we counted every single one of those cells. And what we were assuming was that cavefish would have more red blood cells than their surface fish counterparts. And at this point, we hit our first roadblock because, as you can see, the cavefish did not have more red blood cells. And if anything, some of these populations even trended to have fewer red blood cells than the surface fish. And I was scratching my head a little bit at this one because I very specifically remember I had a sample of blood on a slide and it was stained, and I was looking at them just with my naked eye, And I barged into Dr. Gross's office and I said, cavefish have more red blood cells than surface fish. I know it. And he said, okay, Tyler, run the numbers and we'll see. And I ran the numbers and I was very wrong. And so we were puzzled. We were trying to think about, well, we know that cavefish have a higher concentration of hemoglobin protein. It's not the number of cells. So what could it be? And so the next thing that we thought about was the size. So what we did was we took more blood from our fish. We stained them using a different stain, right GEMSA. And we measured the two-dimensional area of the entire cell, as well as the nuclear area, just to try to see if there were any differences in the different components of the cells. And what we found was that cavefish have much larger red blood cells than the surface fish do. And this gave us a little bit of an answer as to how we were able to achieve a higher hematocrit in the cave populations compared to the surface fish. Now that's the total 2D area. We also measured the nuclear area. And as we would expect, we did not find any differences. And so we're able to attribute the increase in size of the red blood cells to the cytoplasm where hemoglobin is found. And so this kind of was a nice way to wrap up the first story. So what we did first, again, was we measured mean hemoglobin concentration. We found that it was higher in the caves compared to surface. We used the diagnostic tool of hematocrit, and we found a trend higher for all of the cave populations. We felt sure that that had to be explained by an increase in the number of cells, and we found that that wasn't true. They were very comparable between our two morphotypes. But what we found to eventually explain those phenotypes was that the cavefish had larger red blood cells. And so what we can do is we can calculate this metric, MCH, or mean corpuscular hemoglobin, how much hemoglobin is in a single cell. And we find that it's much higher in cavefish compared to the surface fish. So switching gears a little bit, we were very excited by this. And we were like, okay, we have something here. Let's take it a little bit further. So we drew more blood from the fish and we submitted that for RNA sequencing. And what I was really interested in asking here was, do we see evidence of adaptation to hypoxia in gene expression as well as the red blood cell morphometrics? We know that hemoglobin is higher, but in fish and in humans, we have many hemoglobin genes. So is that increase in hemoglobin protein concentration attributed to an overall increase in all of those genes? We also wanted to know, are these phenotypes consistent across life history? And yet another question was, is there evidence? Can we find any evidence for convergent evolution between these different cave populations? So the way that we did this was using the same populations of fish. We used 2 males and 2 females. We extracted whole blood from each of those samples. Owing to the technical requirements from the sequencing core, we actually had to pool those samples so that we'd have enough RNA to sequence. And we were able to send technical replicates of 3 for each one of our populations. The first thing that we wanted to do was we wanted to see, okay, well, if we take the total hemoglobin gene expression, how well does it match up to the protein concentration? And in an ideal world, they match very well. But sometimes the reality is that via post-translational, post-transcriptional modifications, that may not be the case. So that was the first thing that we wanted to check. So what I did was I took the total of all of those hemoglobin genes. So we have the sum expression here, and I overlaid the protein concentration. And what we found was a very high correlation between protein concentration and gene expression. And the big takeaway here was that now we're able to survey those individual family members, because we know that the sum total of expression is roughly equal to, or correlates very well with, the protein concentration. So what we did was we surveyed these individual genes. Now in our species, most of the adult hemoglobin genes are found in this region of chromosome 3. This region is very highly conserved across fish, across mammals, birds. And we know that because on either side of this region, there are many, we refer to them as flanking genes, genes that aren't hemoglobin, but are found in fish, in humans, in every mammal, in birds, in reptiles. And so what researchers are able to do is they're able to find those genes and look in between them for the hemoglobin genes. So here, each one of these blue bars represents a single hemoglobin gene. And what I'm going to show you is the expression values for each one. And the way that we did that was by coloring in circles. So the darker the circle, the higher the gene expression. So by looking at this, we were able to take away a few things. The first one, which I find very interesting, is that this here, the fourth one on the left, is the highest expressed gene, regardless of what population you look at, regardless of morphotype. It's the highest expressed in surface fish. It's the highest expressed in all 3 of our caves. Now, interestingly, I should point out that it's much higher in Tanaha, especially compared to Chica and surface. Now, it's interesting that I say that because one thing that we weren't entirely expecting was this very close relationship in gene expression between the surface fish and the Chica cavefish. And I had to run the numbers on this a couple times because for me, they almost look identical. But I promise you not, I've run these probably more than what Dr. Gross would even like me to do. And I can assure you that they're not the same. But we found this coalition or this grouping between that Pachón cavefish and Tanaha cavefish, and interestingly, the Chica cavefish in the surface morphotype. The other really interesting takeaway for me is that if you look very closely, there are a couple genes that are expressed higher in surface and in Chica compared to Pachón and Tanaha. So especially these 2 genes all the way over here on the right, you can see that those circles are darker in surface and in Chica. Now, we don't know for sure why this is, but my best guess resembles something like those codfish that evolved to live in more variable environments. And the way that I would explain this, and we haven't tested this yet, but I would love to, is that I assume that these 2 genes are not beneficial for the Pachón and the Tanaha environment. And there may be slight differences in how much oxygen Chica has or how much it fluctuates compared to Pachón and Tanaha. And that may explain why their gene expression pattern more closely resembles the surface fish compared to the other 2 cave populations that we surveyed. So another question that we had was, will this pattern hold across life history? So here we're surveying adults only. What if we look in juvenile fish or young fish? And what we found was something pretty similar. So I'm going to show you a very similar figure. This time the circle is going to be divided in half, each half indicating a developmental stage. So on the left half of the circle, the fish are 36 hours old. And on the right side of the circle, they're 72 hours old. And if we look, here we're just surveying surface Pachón and Tanaha, we see something kind of similar, where overall, we see much greater expression in Tanaha and Pachón compared to the surface fish, but it's not all of them. And it's subtle, and it may be a little hard to distinguish from back there, but we see a couple genes, especially here, where the values are actually higher in surface fish compared to Pachón and Tanaha. That was very interesting to me. Another thing that I found interesting was, although the general pattern of having more hemoglobin in the cave populations compared to the surface population holds, the timing of peak expression differed across the two cave populations. So here you can see a good example where in Tanaha, the darker half of the circle is on the right at 72 hours post-fertilization. And for the same gene in Pachon, the darker half is on the left, not the same. Left in Pachon, right for Tanaha. And so it seems that Tanaha may be taking a little bit longer to really increase the amount of hemoglobin that they're expressing compared to Pachon. Now, I was very focused on hemoglobin owing to the previous study that we looked at. But of course, when we run RNA sequencing, we get data for all of the genes in the genome or all of the transcripts in the transcriptome. So I wanted to zoom out and see what other patterns we see between these three cave populations and the surface population. So the way that we did that was we ran an enrichment using gene ontology. So categorizing these genes into their potential functional abilities. And when we compare them to surface fish, we did this for each of our populations, we found some interesting things that were unique in some of the caves, but also shared in some of the caves. So I don't expect you to read all of these terms. So I'll generalize for you that unsurprisingly, in Tanaha and Pachon, many of the terms had something to do with oxygen transport, whether it be oxygen binding or moving the oxygen along. And interestingly, we saw a lot of overlap between Pachon and Pachon and Chica. And these terms largely dealt with immune function. One thing that I wasn't quite expecting was that we did not find any of the same terms when we looked at Tanaha and Chica. They didn't share any of the same terms. Now, one way to explain this is that their environments may just be very different. However, I was a little bit concerned that it could be possible that just the way that these statistics are run, we may be missing something. And so I kind of wanted to just take another approach to see, are there other genes of interest that are expressed much higher or lower in the cave populations compared to the surface fish? And so what I did was I manually did all of this work in Excel, and I found many more genes of interest. And so here on the left, I'm showing you a table of 20 of those genes that have very similar patterns in all three cave populations compared to the surface fish. Now, again, I don't expect you to read all of this, but I will note that on the left side of this table, those are genes that are all expressed much higher in the cave populations compared to the surface. And on the right, they're lower. So I did this for the three populations of interest, but given that we found much similarity between Tanaha and Pachon, and differently Chica and surface fish from the prior study, I also wanted to just look at what might be shared between Tanaha and Pachon, omitting Chica, just to see if we would find anything interesting. Now, what I was not expecting to find was that many of these genes have something to do with a typical response to hypoxia. So while many species aren't adapted to living in low oxygen, most species have some type of response. So you can even think about it in humans where if you're struggling to get enough oxygen, you might breathe more. So there are behavioral responses. If you're running low on oxygen, you're going to start making more red blood cells. You're going to try to find a way to help you secure more oxygen from the ambient environment and deliver it to the tissues that you need. So my first thought was, okay, well, the hypothesis is that cavefish are adapted to living in low oxygen. So perhaps they will express those hypoxia response elements higher. And so what I did, or to help make this easier to see, I've labeled I'm going to label the hypoxia response elements in green and their inhibitors in orange. Now, in a perfect world, we would think that the left side of both of these tables would be filled with green boxes, things that enable response to hypoxia. And that opposite of that, the things that inhibit those responses would all be on the right. So we would expect green on the left, orange on the right. And what I found was that it's actually much more messy than that, where we have many of those orange boxes, those inhibitors that are higher in all three cave populations compared to surface. If we continue, you see a lot of green boxes on the right, where many of those hypoxia response genes are downregulated in cavefish compared to surface fish. And that pattern holds true when we just look at Tanaja and Pachon as well. Now, at first, I was like, oh no, I did something wrong. I mixed up these lists. Something can't be right. But I thought about it, and it turns out that hypoxia response, while it can help you in the short term, it can be very detrimental in the long term. So even if you think about it for yourself, if you are breathing very heavily all the time, that would put a lot of stress on your heart, on the rest of your circulatory system, the other systems of your body, and all of the organs. Additionally, there are even specific types of cancers that hijack this system. They increase hypoxia response as a way to start making more cells unregulated. So they make more red blood vessels. They upregulate angiogenesis. They upregulate erythropoiesis. So they're able to produce more and more cells and produce tumors completely unregulated. So one theory that I have is that cavefish may have found a way to utilize certain hypoxia responses to help them be able to gather enough oxygen from their ambient environment, but to not overdo it, such that they may be causing disease or too much stress. And so if we go back to the questions that I wanted to ask, I believe, yes, we do see evidence for adaptation to hypoxia when we investigate gene expression. We saw a much higher expression of those hemoglobin genes, but we also see very interesting patterns when we look at hypoxia response genes. Interestingly, we found that not all of the hemoglobin family members were expressed higher in cavefish compared to surface fish. In fact, for a few genes, it was the opposite, which I believe may be caused by the differences in their environments, particularly how much oxygen they have and how variable that may be over the course of a day or a month or a year. Interestingly, we did find that the pattern of expression of those hemoglobin genes is very similar in juveniles compared to the adults. And I believe that we do see evidence for convergent evolution, where we saw a very, very tight pattern between Tanaja and Pachon and hemoglobin, but also for many, many other genes, inclusive of response to hypoxia. So to summarize, there are many, many differences between these environments and even within these environments. And I think that it's important for the cavefish community to include dissolved oxygen in the work that we do. But I also think that this is an excellent novel model to study how animals must adapt to lowering amounts of oxygen. The way that we investigated that was first by looking at how much hemoglobin protein they have in their blood, where we found that cavefish have much more compared to the surface fish. One way that that's probably happening is by, that's interesting, by having bigger red blood cells compared to the surface fish. We wanted to investigate this further And one of the ways that we did that was by looking at gene expression, where we found that hemoglobin expression is much higher and it matches very well with the protein concentrations. And we found this very interesting result where I was finding this to be a little bit contradictory to my hypothesis, but to me, I've rationalized it that it might not be such a great thing for cavefish to be upregulating hypoxia response genes all the time. Now, one thing that I do want to remember to include is that all of the fish that we surveyed for these experiments are housed in captive conditions. So they're reared with plenty of oxygen. Now, I think that's a good thing, and I think it's a bad thing. I think that it's very interesting and that it suggests that many of these traits are fixed. So regardless of how much oxygen they may have at their disposal, their programming may require or may dictate an increase in hemoglobin regardless. So that's very interesting to me. However, I think we may be missing large pieces of the puzzle. And I think it would be very interesting to survey some natural caught or fish that are living in the caves, or maybe even just fewer generations removed. But again, you get answers and you just get more questions. So I have a few more questions that we're currently working on to finish up my dissertation. The first one is, how well do they sense oxygen? So we know that cavefish and surface fish have many differences in the way that they sense the things around them. And if you've listened to Dr. Gross talk, or if you were at our demonstration, you'd know that cavefish have more neuromass, sensory neuromass, and they have more taste buds. So one question that I'm interested in is, do they have more oxygen sensors? And so one of the ways that we're going about answering that question is by using an antibody where we're able to label oxygen sensors in the gills of our cave populations as well as the surface fish. And we're finding some preliminary evidence. I'm not willing to say for certain quite yet, so maybe don't quote me, but we're finding that at least Pachón seems to have a much higher density of neuroepithelial cells or oxygen sensors than surface fish do in their gills. So in addition to how well they're able to sense oxygen, I'm also very interested to know how well they can acquire it from the environment, or how efficiently they can do that. So we're taking a little bit of a different approach where we're looking at one of those traits that I showed you earlier. What is the surface area like of their gills? So if you remember from that carp, they're able to remodel their gills and increase the surface area when they need it. So I'm also interested to know, do cavefish have a larger respiratory surface area? And again, don't quote me quite yet, but we're finding that we think, in fact, that they do. So to wrap up, I'd like to thank Dr. Ornelas Garcia for having us. I'd like to thank you all for listening, especially a couple fantastic undergraduates that I've had the opportunity to work with that helped me get this work done. And I'm happy to take any questions.
Tyler, that is an impressive presentation. I'm really, really excited about so many questions that you are opening with this project. It's crazy. But one of the first things that come to my head was just, we have been suggesting that there are differences related with the phylogeny of the group and some that could be really quick adapting to the caves. And based on this, it was really surprising for me to see these differences between Pachón and Tinaja in contrast with Chica. Yeah. Would you say, because if you go to the caves, Chica, it's really, really anoxic. In my opinion, it's one of the most anoxic caves that we have. So it was really surprising for me. And I was just going to your last part of the presentation that maybe since they are so well adapted to these low oxygen conditions could be the reason why they are doing well or relatively less. I don't know. I was wondering if you have a suspicious about this Chica response, like it's contrast environmental conditions?
Yeah, that's a fantastic question. So I think there's a couple things to think about here. The first is that I would be remiss if I didn't say that the chica fish that we have in the lab were acquired commercially. So we don't know the precise genetic ancestry, and we don't know the precise age of the fish. So for example, many of the blood phenotypes are impacted by age. So that could be one possibility. And I wanted to get that out of the way first. I was very much looking forward to having this talk here because I was hoping to discuss some of this with you. One of the things that I have considered is that while we see lower oxygen in caves when we have the measurements, that's only 1 second in time. And so one hypothesis that I have is that the Chica cave may just fluctuate more based on the geography of the cave itself. That's one idea that I have and that I'm curious about. And so I wonder if Chica may be more plastic than surface or than Pachón and Tinaja. So for example, if we were to rear Chica in hypoxic conditions, perhaps they would resemble Pachón and Tinaja more closely. But because we're not, perhaps that's why they resemble surface fish more closely.
Yeah, and in this sense, I think just what we were discussing yesterday about the hybrids conditions of Chica, we can also, yeah, that is true. But yeah, definitely a very interesting result that you show us. And another thing that I was wondering, it was about if it's possible to measure. You have less expression in some hemoglobin related genes, but you also says that the size of the erythrocytes are larger, so maybe can be a compensation. And I don't know how it's easy to compensate that in the analysis, you know.
So in terms of the gene expression, the way that we tried to control for that was we submitted the same mass of RNA for each sample. So for I don't quite remember the technical specifications, but before we sent them for sequencing, we submitted different volumes so that we could try to control for the mass of RNA for each sample. So maybe that addresses that a little bit at least.
And do you think that maybe single cell RNA sequencing could control or balance a little bit of this? I don't know. I was wondering because of the size of the cells.
Yeah, certainly. I think Dr. Gross and I should write a grant when we get back to Cincinnati to do some single cell analysis because I think you're right. I would expect that if you were to control for, like you're saying, the number of cells, it might work itself out a little bit. Because interestingly, the size of the Chica cells much more closely resembled the other caves. So that's kind of another interesting conundrum where the size of the Chica cells resembles the caves, but the concentration of hemoglobin more closely resembles surface fish. So one of the things that we're actually working on right now that I was hoping to be able to include, but we haven't made quite enough progress, is we've done this same analysis again using hybrids, F1s and F2s. Unfortunately, I don't have enough data to say for certain which way it will go, but it looks like, at least from the F1s, we're seeing an intermediate value between Pachón and surface. So there could be some complex genetics at play where the Chica fish that we've surveyed at least may have cave alleles that dictate cell size, but they may have surface alleles that dictate how they regulate hemoglobin genes. It could be that way. It could be very interesting. I'm very hopeful that more members of the community will start to look at these phenotypes because it would be very easy to recapitulate or redo this my entire dissertation just rearing them in lower oxygen. So I think that there are decades worth of research to be done, to continue to be done with this project. Yeah.
Thank you very much.
Thank you. Yeah.
¿Alguien quiere lanzar? Bueno, entonces, si quieren acercar a Tyler, ahorita vamos a tener café y afuera aquí, entonces podemos charlar aquí al rato con Tyler. Muchas gracias.
Muchas gracias.